The next speaker is Elias Pèan. It's my pleasure to introduce him. He's a pharmacologist by training from the University of Lyon and Paris, if I know correctly. He spent a couple of years in pharma at Eli Lilly and Roche, and is now for more than a decade with EMA and is heading the oncology franchise at EMA, if I'm correct. So thanks very much, and you give us your perspective, EMA's perspective on the current status. Thanks, Elias.
Thank you very much, Jörg. So when I was asked to address key pain points from a regulatory perspective before the JCA implementation, I thought of the question of the comparators or lack of comparators, which is an issue that both regulators and HTA face. And even though we are not addressing or looking at it from the same perspective, it is something that we have to deal with. How do I change the slides, actually? This one. Okay. So if we look at the question of control group from a methodological perspective and the definition we have from ICH, it is to separate the effect of treatment from the effect of other factors such as natural course of disease, other medical care, or the patient expectation. And now I'm stating the obvious, but also when we look at the choice of the control group, it can be concurrent or external. And for concurrent control, we can have placebo, the absence of treatment, different doses, or an active control. Now I just wanted to remind of the legal framework for a marketing authorization. So when we review a clinical trial, what we are looking at is trying to establish the efficacy and the safety and to make sure that the potential risks are outweighed by the therapeutic efficacy of the product. So now, if we look at the oncology perspective, as this will be in the scope of the JCA as of January next year, we have our reference document, which is the guideline on the clinical evaluation of anti-cancer medicinal products, which was developed by the Oncology Working Party at EMA and adopted by the CHMP. So when looking at pivotal trials, the standard data that is expected is coming from randomized control trials. And the aim is to, again, establish a benefit-risk profile of the experimental drug in a well-defined indication. And when we talk about the comparator, we should be selected based on best available evidence-based therapeutic options. And what we mean by that is a widely used regimen with a positive benefit risk, which has been documented through randomized trials. So this is what our guideline says. And obviously, there are a number of challenges when we look at comparators. There could be varying preferred regional standards. And for tackling that, we have to look at superiority design or add-on studies. The comparator could also be the investigator choice. Here, to mitigate the challenge of that, the choice should be reported prior to randomization and detailed, if possible, in the protocol. And stratification is also recommended. We also have the question of non-inferiority design, and here it's very important to have a comparator for which we have a good understanding of the efficacy so that we can have well-defined non-inferiority margins. Also, other issues that we face is the absence of reference regimen, and in those cases, best supportive care with the highest level of evidence can be chosen, or the investigator's best choice in the context of a superiority design. And in some situations, single-arm trials can be used, and I will talk about it later in the presentation. Also, one of the challenges is to maintain blinding in the context of cancer trials, and to mitigate that concern, it's important to select a comparator which have a similar toxicity profile, and to also use a placebo in context of an add-on study. Now, there are a number of issues that we have to deal with. One of them is the fact that we sometimes have to assess suboptimal comparators in the context of a marketing authorization. This is clearly not recommended by our regulatory guidelines, but once it is submitted, we have to assess it as part of the marketing authorization application. This is something that we have to acknowledge, that it is inherent to evolving treatment landscapes. So if we look at, for example, the immune checkpoint inhibitors, so the anti-PD-1, anti-PD-L1. A few years ago, they came as a breakthrough. However, it took some time before it became the standard of care, and it has very quickly became the standard of care. However, there was a number of other anti-PD-1, anti-PD-L1 which were developed in the next few years. But by the time they came to the regulators, the treatment with an anti-PD-L1 had become the standard of care. However, at the time of development, it was not. So we have to also deal with that situation. And in that context, we have used the same standard of evidence that was for the first ones and was replicated in the next one. So we have accepted the add-on design versus chemotherapy, for example. That was the same as the one used for the first ones. And this is the last point on this slide, the importance of having a superiority design in those circumstances. Now, in all other situations, we also have to deal with uncontrolled trials. The situations in which this can be acceptable is limited to those where randomized control trials are not feasible or difficult to conduct. This isn't the case for orphan conditions or very rare conditions, or where we are in a setting where there are no or limited treatment options. However, the choice of such design to support the pivotal, to support the marketing authorization should always be duly justified when supporting a marketing authorization application. And there are a number of challenges that you are aware of with single-arm trials. It's the isolation of the treatment effect, it's important to understand the natural course of the disease in those settings. Also, the assessment of safety is also a challenge if you don't have a comparator to understand what is the safety profile linked to the disease and what is linked to the product itself. And there are a number of biases. I will just mention the selection bias, which is clear for uncontrolled trials. And all these elements have been thoroughly discussed in a reflection paper on establishing efficacy based on single-arm trial, which was published by EMA a few months ago. Now, if we look at single-arm trials in the specific context of oncology, here, same elements. It is not recommended by our guideline. It needs to be, it can be used in very specific context where we have rare conditions, last line with no available therapies. The choice of endpoints is very important. For oncology trials, we look at response rate and duration of response because we need to have an endpoint which reflects the true activity of the drug. And for example, the shrinkage of a tumor cannot happen spontaneously. So this is for sure connected to the activity of the drug. We don't use time to event endpoints because we consider that there are other factors that will contribute to a prolonged overall survival or PFS, for example. It's important to understand the natural course of the disease, so that's why it's always useful to have a historical control. And there is a threshold of activity that is expected, and I will go more in details in the next slide about that, because there is a certain level of activity under which the uncertainty is too high to conclude on a positive benefit risk. Also, what should happen systematically if a single-arm trial is considered for submission to support a marketing authorization is to go for scientific advice. And in many cases, when a single-arm trial has been submitted, it goes through specific regulatory pathways. So we have the conditional marketing authorization, which has to be complemented post-approval with confirmatory data, and I will also mention a few words about it later in the presentation. So in this slide, there are a number of products that have been approved on the basis of single-arm trial. So all of them are in non-small cell lung cancer, in biomarker-targeted indication, and you can see the response rate that was reported in the supporting trial. It goes from 37% to 73%. And here what I wanted to mention is the case of Exkivity, mobocertinib. So this was a product that was intended for the treatment of non-small cell lung cancer with EGFR exon 20. In that case, the response rate was 28%, so much lower than what has been approved so far. The CHMP was not convinced about the positive benefit for risk for that product. The company withdrew the application. It was approved by FDA in 2021, and 2 years later, when the confirmatory trial was finalized and showed that actually it didn't meet the primary endpoint of PFS, the product was withdrawn. So that's where also there is, with single-arm trials, this question of threshold, which is very important to understand or characterize. Also, in this slide, I'm repeating what I've said before. So for marketing authorization, the legal framework is establishing efficacy and safety, and that potential risk outweighed by the efficacy of the product, so the benefit risk, which needs to be positive. And I'm putting it side by side with the legal framework of the HTA assessment, which is to determine the relative effectiveness and focusing on the added value compared to other existing therapies. So we are really asking different questions, and this is something that I want to make also quite clear. However, there are situations where we regulators do assess added value. One of them is the conditional marketing authorization that I mentioned earlier. This is an approval which is based on a data package which is not considered comprehensive. It is restricted to specific indications, seriously debilitating, life-threatening, emergency situations, public health threats, and orphan medicines. Here, what needs to be established is the unmet medical need, and that the product is expected to bring major therapeutic advantage over existing therapies. And and the marketing authorization is subject to a specific obligation, which is a confirmatory trial, most often a randomized control trial, if the initial approval was based on a single-arm trial. Here, just wanted to mention the example of Tagrisso, osimertinib, which was also mentioned in the slide from Professor Nistico. This is a product that was initially authorized in a very small patient population of non-small cell lung cancer with EGFR mutations who developed a specific resistance that couldn't be overcome by other treatments. So this was the initial approval based on a single-arm trial, but there was a number of clinical development that followed, which were actually, one of them was a condition to the marketing authorization, and they were all very successful, and this product has become the standard of care now in the treatment of non-small cell lung cancer with EGFR mutation. Another situations where we do assess added value is the context of the orphan marketing authorization. This is also restricted to a life-threatening or chronically debilitating condition, which affect not more than 5 in 10,000 people. There is a reward coming with this marketing authorization, which is a 10-year market exclusivity. And here, again, there is a requirement to demonstrate significant benefit over existing therapies. And we had the recent case of Viloy, which was approved in HER2 negative gastric or gastroesophageal junction adenocarcinoma with a specific biomarker, which is Claudin 18.2, which for the time before this product didn't have any other product approved for that specific biomarker. And the last one I wanted to mention where we do assess added value is the additional year of market protection. This is in the context of extension of indication. And it's when a new indication brings significant clinical benefit compared to other existing therapies. So significant clinical benefit could mean improved efficacy, improved safety, or major contribution to patient care. And there, for example, if there is an oral administration when all the other existing therapies are IV, that could be considered as a major contribution to patient care. So in this, the message I wanted to convey is that despite we are answering different questions, there are commonalities, and actually the same data could still address the needs of both assessment. Reason why the parallel EMA HTA scientific advice is something very important to try to streamline as much as possible the evidence generation that could address both needs in anticipation of the joint scientific consultation. And a few elements that I want to highlight here is that obviously you're aware probably that there is an implementing act which is open for public consultation on the joint scientific consultation, and the deadline for comments is the 29th of October, if I remember correctly. And what will try to be achieved in this implementing act is to have timelines that are synchronized between the joint scientific consultation and the scientific advice working party from EMA, that the templates are as much as possible aligned also between the HTA coordination group and EMA, and also that the parallel EMA HTA joint scientific consultation process is supposed to be the default format. So a few messages I wanted to make sure I convey today is the focus of the marketing authorization is the benefit risk, so that the benefit outweighs the risk for a specific product. This is usually demonstrated by randomized controlled trial, but not always. Added value can be assessed by regulators in specific situations, and also to highlight that the same data sets can support both benefit risk and added value discussion, but for that there is a need to have a discussion upfront in the context of scientific advice and later joint scientific consultation. And thanks for your attention.